Hi there, my name is Viktoria Schuster, and I will be presenting my poster titled Encoder-less Generative Modeling of Transcriptomics Data for Improved Representations. I am a third-year PhD student at the University of Copenhagen. This work has been done by myself, my supervisor and group leader Anos Kroh, and one of our postdocs, Inigo Prada-Luengo. We work in the interdisciplinary field of machine learning methods for biological and health-related problems. Biological data is vastly growing, which makes it suitable to investigate using machine learning methods. Since it is often complex, machine learning is a perfect fit. Generative modeling can help us uncover the underlying mechanisms of this complex data. However, many of the available methods show some serious limitations for this application. Because of these limitations, we aim to develop a generative model that is first of all simple, but can model more complex underlying distributions. Further, we wanted our model to be better suited for complex, high-dimensional biological data that often comes with few samples. The result is the Deep Generative Decoder. Encoders are practical ways to map from data space to latent space, but they require more data to be well determined than decoders. Coming from the manifold approach and applying maximum a posteriori estimation instead of maximum likelihood, we can remove the encoder and learn representations directly. For the model to be generative, we also need a prior distribution over latent space. Because we do not require the Kullback-Leibler divergence, it is pretty trivial to use any differentiable distribution here. We chose to plug in a Gaussian mixture model in order to be able to fit more complex spaces than a standard Gaussian, as is the default in many other models. Optimization in our model is based on the reconstruction of the data and the match between representation and Gaussian mixture model. The first is given by the conditional probability of data X given representation Z and decoder parameters theta. The latter is given by two terms. Firstly, we need to calculate the probability of representation Z being drawn from the parameterized Gaussian mixture model phi. Secondly, the GMM is conditioned on priors over its means, covariance matrix, and mixture weights. When embedding and predicting new samples, we get representations by first finding the best GMM component for each sample, and then performing stochastic gradient descent for a number of steps. Our model presents three distinct benefits, which make it very suitable for complex biological data. Firstly, we can learn higher quality representations without the encoder. We have demonstrated this on simulated data where the decoder alone can recover the ground truth, whereas the autoencoder might need an incredibly large amount of data to do so. Secondly, our method provides an integrated clustering of data with more controlled generative capabilities. Lastly, our method is highly data efficient, which becomes extremely apparent in our application to high-dimensional data with few samples, which I will introduce now. For our application, and because we are very interested in gene expression and regulation, we chose both bulk and single-cell gene expression data. On bulk data, we observed good clustering of tissues and can perform differential expression analysis with much higher precision than DSEQ2. This work was led by Inigo Prada-Luengo. On single-cell data, we have demonstrated that our model achieves great unsupervised clustering and can identify subclusters not found in data labeling. This feature can simplify people's workflow because the clustering comes with the model and removes the necessity to perform additional graph and clustering computation, while not being slower than comparable models. In our comparison, we have focused on SCVI, which seems to be the most widely applied and successful generative model for single-cell expression data. We are happy to announce that we could drastically increase the scale of datasets that can be modeled with our approach. Theoretically, there is nothing to gain in the limit of very large datasets. We do still perform on par, though, with SCVI for a 1 million cell mouse brain dataset. However, where our method especially shines is in regimes of very small sample sizes. As you can see, our model, which we call SCDGD, clearly outperforms SCVI on datasets smaller than 3,000 cells and can perform very well on datasets as small as 500 cells. SCDGD has been accepted for publication in Oxford Bioinformatics and is available as a Python package on our GitHub. Please check it out if you're interested. If you would like to read our papers, you can find them using these QR codes. We are also working on multimodal modeling. I will soon publish my preprint on multiomics data, which I worked on together with the Teichmann Lab at the Wellcome Sanger Institute. Keep tuned. Thank you very much for listening to my poster presentation. If you have any questions or would just like to reach out, come find me at my poster or send me an email. Have a lovely day.